Look around a family gathering, and you will usually spot a mix of physical features. A child might have their mother's nose, but their father's eye color. We can clearly see that these physical traits are distinct. But back in the 19th century, the scientific consensus was very different. Biologists believed in a concept called the blending theory. They thought a child's traits permanently mix together, just like pouring yellow and blue paint into a bowl to get a uniform green. There is a major mathematical problem with this idea. If every generation kept blending together into an average, eventually all variety would vanish. An entire population would look exactly the same, like a single shade of dull gray. Since we don't see that happening in the real world, the paint mixing theory had to be wrong. During the mid-1800s, 3 men, working entirely on their own, were about to dismantle that theory. Finding the instructions for life took time. 3 separate researchers worked in isolation, unaware that their individual findings would eventually reveal a single, clear picture of how we inherit our traits. Charles Darwin spent years observing wildlife across the globe. He carefully documented how animals of the same group had slightly different physical features depending on the specific environment they lived in. He realized that species are not fixed in place. Over many generations, they change to survive better in their surroundings. If an environment has hard-to-crack seeds, a bird species with a beak that gradually thickens over time will get more food, survive longer, and have a better chance of having offspring. He called this overarching process natural selection. It proposed that all species of life descend from common ancestors, slowly adapting to their habitats over vast stretches of time. But Darwin faced a massive gap in his own logic. He could see that an animal passed its successful traits to its young, but he had absolutely no idea how that physical transfer actually happened. He had observed the motivation for evolution, the survival of the fittest, giving science the why, but the biological mechanism, the how, was missing entirely. Around that same time, a monk named Gregor Mendel was working in an abbey garden. He spent his days meticulously crossbreeding thousands of common pea plants to track how they inherited physical features. Looking at this table of pea characters from Mendel's experiments, we can see the specific variations he tracked, like flower color and seed shape. By counting these variations over generations, he proved that traits do not blend together like liquids. They are handed down as distinct, separate packages. A purple flower crossed with a white flower didn't make a pale purple flower. It made flowers that were either entirely purple or entirely white. He also discovered that some of these packages overpower others. Continuing to look at Mendel's data, we can see the traits categorized into 2 groups. The column on the left shows dominant traits, like round seeds, which will physically appear even if paired with the less powerful recessive traits on the right, like wrinkled seeds. Mendel called these packages heritable factors. Today, we know them as genes. Through careful counting, he had successfully worked out the strict mathematical rules for how these packages move from parent to child. Yet his tools were limited, and he couldn't actually see these packages inside a cell. He had decoded the logic of our biological instruction manual without ever knowing what the ink or the paper was made of. The answer to what that material was made of came from a doctor-turned-researcher named Friedrich Miescher. He was curious about how cells were chemically built, specifically wanting to know what their large centers, the nuclei, were made of. To get enough cell material to study, he used an unusual method. He collected used bandages from a local hospital, bandages originally used on soldiers from the Crimean War, and scraped the pus off them to gather large quantities of white blood cells. By applying chemical washes, he broke open the cells and stripped away the outer layers, leaving behind just the dense nuclei from the center. Looking at this flowchart, we can see his breakthrough at the bottom right. He isolated a new, acidic substance rich in phosphorus and nitrogen. This was a completely novel substance. He named this substance nuclein because it came from the nucleus. Today, we know it as nucleic acid. It is the physical material that makes up our genetic code. Miescher had found the actual physical molecule of life. He gave us the what, even though he had no idea his discovery carried Mendel's invisible traits or powered Darwin's engine of evolution. The historical irony is that Darwin, Mendel, and Miescher were all doing their work right around the same time. Yet their massive discoveries were largely ignored or misunderstood by the wider scientific community for decades. It wasn't until the early 1900s that new generations of scientists started pulling these separate bodies of research out of the archives and placing them side by side. When they did, they realized that these three isolated men had actually been staring at the exact same biological process from three different angles. Miescher's isolated nucleic acid, what we now call DNA, was the physical form of Gregor Mendel's invisible packages of traits. The rules Mendel counted in his peas were dictated by the chemical structure Miescher found in his test tubes. And the successful passing down of that DNA from one generation to the next is the exact engine that allows species to adapt to their environments, answering the missing question in Darwin's theory of natural selection. Science often works like this. Individual pieces of evidence, gathered over long periods, eventually unite to reveal the deepest secrets of our existence.